Welcome back, students. From previous lessons, we know that in the vascular system, arteries, capillaries, and veins are connected in series to achieve the physiological functions of blood transport and material exchange. The ultimate goal is to deliver nutrients and oxygen to tissues and remove metabolic waste products. Therefore, the microcirculation, as the site of material exchange, plays a vital role in maintaining tissue cell metabolism and internal environment homeostasis. We refer to the blood circulation between arterioles and venules as the microcirculation. As shown in the diagram, a typical microcirculatory unit includes arterioles, medarterioles, precapillary sphincters, true capillaries, thoroughfare channels, arteriovenous anastomoses, and venules. Naturally, the composition of the microcirculation varies among different organs and tissues due to their differing structures and functions. For example, the microcirculation in human fingernail fold skin is relatively simple, with only looped capillaries connecting arterioles and venules. In contrast, the microcirculatory structure in skeletal muscle and mesentery is considerably more complex. Let's first examine the functions of the various components that constitute the microcirculation. The starting point of the microcirculation is the arteriole, whose wall has a complete layer of smooth muscle. Contraction or relaxation of the circular muscle in its wall can significantly reduce or enlarge the lumen diameter, functioning as the main gate controlling microcirculatory blood flow. Arterioles branch into medarterioles with finer diameters, whose walls contain only a single layer of smooth muscle. Each medarteriole supplies blood to one to several true capillaries. At the origin of each true capillary, there are usually 1 to 2 smooth muscle cells forming a ring-like precapillary sphincter. Its state of contraction determines the blood flow into the true capillary, acting as a distribution gate in the microcirculation. True capillary walls lack smooth muscle and consist of a single layer of endothelial cells, enveloped by a thin basement membrane, with a total thickness of only about 0.5 micrometers. Minute gaps exist at the junctions between endothelial cells, forming channels connecting the inside and outside of the capillary. Hence, capillary walls are highly permeable. They are the primary site for material exchange. Blood from the capillaries enters the veins via venules. The smallest venules have a diameter not exceeding 20 to 30 micrometers. Their walls lack smooth muscle, and they also function as exchange vessels. Larger venules possess smooth muscle and belong to the postcapillary resistance vessels, acting as the posterior gate. Through their contraction and relaxation, venules can influence capillary pressure, thereby affecting fluid exchange and venous return. From the schematic diagram, we can easily see that there are many branching paths in the microcirculation. Therefore, based on the flow paths of blood through the microcirculation, we can categorize the microcirculatory pathways into the circuitous channel, the thoroughfare channel, and the arteriovenous shunt. The first pathway is the circuitous channel. The circuitous channel refers to the microcirculatory pathway where blood flows from the arteriole through the medarteriole and precapillary sphincter into the true capillary network, finally converging into the venule. This pathway is named for the numerous, winding, and tortuous true capillaries, coupled with thin walls, high permeability, and slow blood flow. It is the primary site for exchange between blood and tissue fluid, hence also called the nutritional channel. True capillaries in different parts of the same organ or tissue open alternately. Furthermore, the same capillary alternates between being open and closed, controlled by the contraction and relaxation of the precapillary sphincter. At rest, approximately 20% of the capillaries are open at any given time, matching the metabolic needs of the organ or tissue at that moment. The second pathway is the thoroughfare channel. The thoroughfare channel refers to the pathway where blood flows from the arteriole through the medarteriole and the thoroughfare channel into the venule. The thoroughfare channel is the transitional part of the medarteriole, where the smooth muscle in its wall gradually diminishes and disappears. The thoroughfare channel is commonly found in skeletal muscle. Compared to true capillaries, it is shorter and straighter, has lower flow resistance, faster velocity, and is often in an open state. Its main function is to allow a portion of the blood to rapidly enter the veins via this pathway, ensuring venous return. Additionally, a small amount of material exchange with the tissue fluid can also occur in this pathway. The third pathway is the arteriovenous shunt. The arteriovenous shunt refers to the pathway where blood flows directly from the arteriole through the arteriovenous anastomosis into the venule. The walls of these vessels are relatively thick, with a well-developed longitudinal smooth muscle layer and abundant vasomotor nerve endings. Blood flow velocity is high, and they have no material exchange function, hence they are also called non-nutritional channels. Their function is to participate in body temperature regulation. These shunts are primarily distributed in the skin of the fingers, toes, lips, nose, and within certain organs. They are usually closed, which helps conserve body heat. When the ambient temperature rises, the arteriovenous anastomoses open, increasing skin blood flow and facilitating heat dissipation. Early intervention is one of the key principles in rehabilitation. In rehabilitation therapy, a significant portion of patients are bedridden. Prolonged bed rest leads to localized tissue pressure, which ultimately causes impaired blood circulation, local ischemia, and results in varying degrees of ischemic necrosis of the local tissue. This is a pressure injury, bed sore. Early rehabilitation intervention can not only effectively prevent complications caused by bed rest, such as pressure injuries and muscle atrophy, but also improve abnormal standing and walking postures in the mid to late stages of patient recovery.